Hello, everyone. Thanks for attending. My name is Gerry. I'm one of the field application scientists for 10X here in Australia. I'm based in Melbourne, and I focus mainly on the single cell part of the portfolio. So today we will be doing a site prep presentation. So kind of high level overview of kind of the workflow, the things that you need, and kind of general considerations about sample prep. About the Chromium single cell gene expression solution. So this is going to be mainly focused on the 3' gene expression technology. I will discuss a little bit about the feature barcode technology, but of course there are many 10X assays. So if you are interested in any of the other ones, please let us know. But today we'll just be focusing on the simple 3' gene expression one. Okay, so the aim of today is to give an overview of the Chromium single cell gene expression solution. We'll talk about considerations for sample prep and sequencing, required materials and equipment. We'll touch a little bit on the Cell Ranger software that we use for analysis, and then we'll discuss a little bit about training next steps. Of course, there's another presentation next week as well about the workflow. So the training goals, the overall training goals are to kind of discuss the critical concepts for single cell transcriptome analysis. So describing the relationship between input cell quality and output data quality. We'll review some best practices for cell handling and counting, so sample prep basically. And also review some correct chip handling and loading processes. This will be more, we'll focus more on this in the workflow part of the presentation next week. We'll also talk about executing the gene expression workflow steps. So this is mainly going to be talked about next week, but we'll touch on some of the key points briefly today. And we'll also introduce the fundamentals of gene expression data analysis. So reviewing key steps in Cell Ranger and the usage of Loop browser. But today it's going to be mainly this first portion. Okay, so I'll get straight into it. The Chromium single cell gene expression solution overview. Okay, so the Chromium single cell gene expression solution with feature barcoding technology enables gene expression analysis at the single cell level. So feature barcoding technology provides the ability to simultaneously assess CRISPR perturbations and also protein abundance in the case of cell surface protein. Cell surface protein applications. So this solution is ready to use. It has a robust workflow and is compatible with the demonstrated protocols for multiple sample types, including cell lines, primary cells, and also dissociated fresh tissue. So this particular workflow, the 3' gene expression workflow, is compatible with both whole cells and nuclei. And the solution also includes turnkey analysis and visualization software. So you can see in this image here, on the left here, we have the Chromium controller instrument. This was our first instrument. And on the right here, we have the Chromium iX instrument, which is our latest instrument for single cell analysis. So the only difference in these instruments are the ability of the Chromium iX to perform some assays that the Chromium controller can't. But in the case of 3' gene expression, it can all be performed on both instruments. And in the back here, you see an image of our Loop browser, which is our data visualization software. Okay, so very high level overview of the workflow. The user, so that's you guys, provide us with cells or nuclei in a single cell or nuclei suspension. These are then inputted into the instrument, be it the iX or the controller. I believe you guys have the controller there at the KMB. On the controller, this is where the single cell partitions are generated. So these are the gems. And then following the gem generation and a bunch of library construction steps, you'll make your final libraries, which will be sequenced on usually an Illumina sequencer. The output from these, the sequencing can then be input into our Cell Ranger pipeline, which is our data analysis pipeline. And outputs from the Cell Ranger can then be taken on for further analysis in custom R packages, or taken into our Loop browser software, which is a data visualization software where you can perform some analysis as well. So in terms of the single cell 3' gene expression, the things that you need are the single cell 3' kit, the ChIP-G single cell kit, the dual index plate TT set A, and then you'll also need, in addition to those, these other products listed down here for feature barcoding. So in terms of, for example, if you wanted to do surface protein, you could use the 3' barcode kit here. And then if you wanted to do cell multiplexing, you could use our CellPlex kit set A here. Okay, so this is what is happening in kind of the workflow in both the instrument and then further steps downstream as well. So the workflow for generating a single cell 3' library combines barcoded sequences with high resolution partitioning of the inputs into an emulsion on a microfluidic chip. So during this workflow, your single cell or nuclei suspensions are combined with a master mix and then loaded onto the chromium chip, along with the 10x barcoded gel beads. So these are the gel beads here, and these are your cells plus enzyme here. And then you'll also have another well sample with partitioning oil, and this is also the well where you collect your partitioned gems from. So the chip is then placed on the chromium instrument where the gems are generated. Most of the gems will contain a 10x gel bead, while only a small subset of them will contain both the 10x bead and a single cell or nuclei. After these gems are generated, the gel beads dissolve and the cells lyse, and this releases the oligos from the gel beads and the mRNA from your cells into solution. Within the gems, a reverse transcription reaction then occurs, which converts the mRNA and or your features of interest into 10x barcoded cDNA and feature barcode DNA. So after this barcoding, your fragments are then pulled together into a final library construction step, which is performed in bulk to generate your final library that's then ready for sequencing. The final library constructs are then sequenced, and the 10x barcodes are used to associate individual reads back to individual partitions and to each individual cell. The resulting output you get is a digital single cell gene expression profile with information on your feature of interest, such as CRISPR perturbations or cell surface protein expression. So what's happening inside the instrument, inside each of these gems? So inside each of these gems, you'll have a single cell gel bead. So each gel bead contains many copies of 3 types of oligos, each having the same barcode, 10x barcode here shown in green, but each having a unique UMI or unique molecular identifier. So in terms of capturing your mRNA, the first oligo is used. So this particular oligo has a TrueSeq read 1 handle. It then has the 10x barcode. This is a 16 nucleotide barcode, and there are approximately 3.6 million defined barcode sequences. You then get a 12 nucleotide unique molecular identifier. And finally, your poly-DT, which is what captures the poly-A tail on your mRNA. To enable feature barcoding technology, there are also 2 additional oligos present on the gel beads. These contain capture sequences for your features of interest. So these features may be oligonucleotide conjugated antibodies for cell surface protein profiling, or small guide RNAs for CRISPR experiments. Both of these oligos contain a 22 nucleotide partial Nextero read 1 sequence, as well as the 10x barcode and UMI sequences. However, there are 2 different 22 nucleotide capture sequences present, either capture sequence 1 or capture sequence 2. And these sequences are the reverse complement to the sequences present in your DNA or RNA based feature. So depending on whether it's protein or CRISPR, they would be captured with either capture sequence 1 or capture sequence 2. Okay. So how are the gems actually generated? The single cell solution utilizes the Chromium Next Gem Chip G single cell. So each different 10x assay uses a different chip. In the case of 3' gene expression, we're using chip G. So this particular microfluidic chip enables partitioning of individual cells with 10x barcoded gel beads into droplets. So the lanes on this chip run vertically. So as you can see, there are 8 channels on this chip that can be run in parallel on this single-use chip. So each channel can accommodate between 500 and 10,000 cells per sample. And we have tested up to 30 micron cell diameters. This system provides you also with the flexibility to run less than 8 samples per chip if you want, as long as the unused lanes and wells are filled with 50% glycerol. So these chips cannot be used more than once, regardless of whether you're running 1 lane or 6 lanes or 8 lanes. And the partitioning on the Chromium instrument takes about 18 minutes. So during partitioning, up to about 65% of the cells are captured. So this means that if you were loading approximately 16,000 cells for one experiment, you would yield data from approximately 10,000 cells. So while most of the occupied gems will contain a single cell, there is a statistical probability that some of these gems contain more than 1 cell. And the expected fraction of droplets with more than 1 cell increases linearly with the number of targeted cells. So for 1,000 cells, every 1,000 cells, the multiplet rate is 0.8%. So this, in addition to the fact that the wells don't run to completion, means you're getting, adds up to this kind of 65% cell processing efficiency that you're getting. Okay, so this is kind of a flow chart of the workflow. The workflow steps include cell or nuclei preparation, here with a very optimistic 1.5 hours of prep time. Obviously, this can be longer depending on your sample type. So yeah, take this with a grain of salt. Gem generation and barcoding, and then takes about 1.5 hours. Then you move on to your post-gem RT cleanup and your cDNA amplification, which takes about another 1.5 hours. And then finally, you'll do a final cleanup, which takes about 30 minutes. You'll then take your pellet to take forward to gene expression library construction, which takes about 3.5 hours, after which you can sequence this library. The supernatant from this first cleanup you take is where your features are. So in terms of if you wanted to look at cell surface protein expression, this is where that information is. So you would collect the supernatant and construct a separate library that you can then construct in parallel with your gene expression library and then take on for sequencing. I'll now move on to considerations for sample prep and sequencing. Okay, so we have cell preparation guides on our website that you can look at. There are different cell preparation guides for different sample types that we've tested in-house. However, there's also a general cell preparation guide, which just contains some general information. Obviously, everybody's samples are very different, and each sample has its own little quirks and considerations that you need to think about. But some of these general practices are very good things to keep in mind, especially when we're considering the microfluidics on the 10x chip. So this particular guide includes best practices and general protocols for washing and resuspension, straining your cells, counting your cells, optimal cell concentrations to work with, and also working with very limited cell suspension. So that's less than 100,000 total cells. So in terms of the cells that you can work with, the workflow accepts both flow sorted and primary white blood cells. These can be treated like cell suspension lines, or you can work with other cell types as well, of course. And as I said already, there are many protocols that help you deal with different sample types. Okay, so general cell handling recommendations. So to perform the single cell gene expression assay, we require a fully dissociated single cell suspension to be input onto the chip that goes into the Chromium instrument. So this means minimizing the presence of cellular aggregates, dead cells, and also non-cellular nucleic acids, and also potential inhibitors of reverse transcription like EDTA. So this is all critical to obtaining high data quality. Obviously, we're working with microfluidics, so very narrow channels. So we want to avoid aggregates because these can cause clogs on the microfluidics. Dead cells can give out nucleic acid that can cause cells to aggregate together, again, causing potential clogs. And also, because we are doing a reverse transcription reaction inside the gems, we want to also avoid those reverse transcription inhibitors like EDTA. So you can work with suspension cell lines, bead enriched, and also flow sorted cells with this particular assay directly after washing and sorting and washing. The cell quality is very important. So ideally, we recommend that you input cells onto the chip that have viability of more than 90%. Obviously, this isn't always possible. So sometimes you may work with samples of lesser viability. We find that when you have a high fraction of non-viable or dying cells, this will decrease your recovery, since the presence of ambient RNA and cellular debris can negatively impact performance and also your data at the end. Obviously, you want to be sequencing healthy living cells, and yeah, dying cells can give out ambient RNA, which can then contribute to the background in your data, and also, worst case, even cause aggregates of cells to come together and clog the instrument. So it's very important to treat your cells very gently and to minimize cell lysis and loss again. So when cells lyse, again, they will release ambient mRNA and contaminate other gems. We recommend that you wash your cells twice before loading onto the chip with a wide bore pipette tip to remove any ambient RNA and contaminants. And the wide bore is to make sure that your cells are being pipetted very gently, and you're not putting them through a very narrow kind of pipette bore where they could potentially lyse. We also recommend that you wash and resuspend in PBS plus 0.04% BSA to minimize cell loss during handling. This concentration of BSA can be adjusted, but this is a good starting point. Okay. Next, it is also very important to remove any debris and aggregates. So using a cell strainer to remove these from your washed cells is very, very important, since the presence of these cellular aggregates and debris and/or fibers can result in both inaccurate cell counts and also clogging of the microfluidics. So gem generation occurs in microfluidic channels that are narrower than a typical human hair, so less than 100 microns. So the presence of any debris or any large aggregates can cause clogs or wetting failures, which is where you fail to generate a proper emulsion. We like you to quantitate your cells accurately before loading onto the system. So as I said previously, approximately 65% of your cells are loaded. 65% of the cells that you load will be recovered. So to maximize the likelihood of achieving the desired recovery target, the optimal input cell concentration is 700 to 1,200 cells per microliter. Our recommended range is 500 cells up to 10,000 cells recovered, or up to 30,000 if you're performing cell multiplexing with our CellPlex product. So if you under or overload the instrument, this can impact application performance. So it's really, really important to make sure you get accurate cell counts, because if you underload, then you are not you're not going to be performing the right number of amplification cycles, for example, to amplify your cDNA product. And if you overload, that can mean potentially clogging the instrument as well, clogging the chip, sorry. Okay, and finally, your cell suspensions should always be kept on ice wherever possible, and you want to proceed with cell loading immediately after your sample prep. So you want to keep your incubation times to a minimum, and you want to move on quickly. Obviously, we don't want you to rush, but we want you to move on, so that the cells aren't sitting around for a long period of time where they can start to die off. Some cell types are more fragile than others, so cell viability may decrease significantly if they're not processed and loaded immediately. So it's important to move on quickly at a good pace. Okay, so at the end of everything, once you have constructed your libraries are going to look like this. And these libraries are compatible with Illumina sequences. So for a single cell 3' gene expression library, we recommend a minimum of 20,000 read pairs per cell. I'm not going to go into the details of this library construct too much today. I'll be going through it more in the workflow presentation next week. So these libraries are compatible with any of the Illumina sequences, basically. So this is the library construct. And yeah, so for read one, we recommend a read length of 28 cycles, 10 each for the I7 and I5 index, since these libraries are dual indexed. And finally, we recommend 90 base pairs for your actual insert of interest. The sequencing depth for typical samples, we recommend 20,000 raw read pairs per cell. So this is for a typical sample. Obviously, different cell types have different RNA content, so you may need to sequence more deeply or not as deeply for your cell type of interest. So, in this graph, you can see, for example, that HEC293 T cells have a very high RNA content, so you need to sequence more to reach saturation, whereas PBMCs do not contain that much RNA, so you don't need to sequence as much to reach that saturation point. Also, given the variability in cell counting and loading, extra sequencing may be required if your cell count is higher than you anticipate. So this is another reason to really make sure your cell counts are very, very accurate. Okay, now moving on to the required materials and equipment. So this is what the kits look like. So the single cell gene expression solution with feature barcode technology. So this is what you will get in your kit. You'll get the next gem single cell 3' gem kit. If you're doing CellPlex, you'll get a CellPlex kit. You will get your gel beads. And if you're doing feature barcoding, you'll get a feature barcoding kit, as well as your library construction kit, which applies to all of these. Do note that there are Dynabeads that come with these kits that come in a little envelope, so don't chuck these out, keep them. You'll also get partitioning oil, recovery agent, your actual chips and gaskets that go onto the instrument here, and you'll require different index plates for whether you want to do just single cell gene expression or single cell gene expression plus feature barcoding or CellPlex as well. The important thing to note is that things are either stored at -20 or at ambient temperature. But the important thing to note is that the gel beads absolutely need to be stored at -80 degrees. If you store them at -20, they will clump together and will be useless. So please, when you receive your gel beads, put them at -80. And similarly, so the Dynabeads go at 4 degrees. Everything else is either ambient or -20. Other things that you will need plastics. So this particular table contains plastics that have been validated by 10x Genomics and are highly recommended for the assays that we provide. So substituting some of these materials may affect system performance. In terms of tubes, we recommend USA Scientific, Eppendorf, and Thermo Fisher PCR 8-strip tubes. And in terms of pipette tips, we recommend Rainin LTS tips, also Eppendorf ones. The reason why we are so specific about the plastics that we recommend is that in terms of the strip tubes, certain plastics are not compatible with the emulsions that we generate and can cause them to break prematurely. So please do stick with the ones that we recommend. In terms of the actual pipette tips, it is also very important, since some of the other brands that we've tested in-house do not have the same kind of QC procedures that the Rainin ones do and the Eppendorf ones do, and they can actually have a very jagged wall inside, which can cause your cells to die and lyse. And also some of them do give off little bits of microplastics that you can't see under the microscope that can then go on to clog the chip. So when working with your samples, do try to stick with the plastics that we recommend. Additional things you need. So for quantification and quality control of your cDNA and the library that you eventually generate, you will require either a bioanalyzer, a tape station, or a lab chip to look at your cDNA, to quantitate your cDNA and also assess its quality, and also look at the quality of your final library as well. So here's a list of the things that we recommend for doing that. I will send these slides to you guys afterwards, so no need to jot any of this down. You will get copies of these slides. And finally, you need to have a thermocycler that can support uniform heating of 100 microliter emulsion volumes. So any of these thermocyclers are recommended, and we also have other ones that we recommend as well. So please let me know if you have something that's different to this, and we can discuss whether it's compatible or not. Okay, so now moving on to the analysis portion. So the Cell Ranger software is the software that we use for analysis. Basically, the kind of workflow is that you'll have your cells, which are then partitioned into gems, which are the gel beads in emulsion. These gems are where your single cells are partitioned and from which you generate libraries from. The libraries are sequenced. After sequencing, you'll get your BCL files, which you will put input into Cell Ranger, our analysis pipeline. This will then generate your FASTQ files and also do all the counts. And eventually, it will the Cell Ranger will output a bunch of different files, including the featured barcode matrix band file, the run summary, as well as the C-loop file, which is the file that you input into our loop browser to visualize and play with your data. Yeah, so this is kind of a repeat, but basically Cell Ranger will spit out a bunch of outputs that you can then take onto downstream analysis in Python and R. The most important things to take a note of is the HTML run summary. So this summary contains the run metrics and basic visualizations for your data. And this is what we will look at to assess the quality of your particular sample and run. And this is also what we will use to help you troubleshoot any issues that come up. So this contains a lot of useful metrics that we can use to determine whether your sample quality was good, whether you had any kind of failures during your run, and how the sequencing went, basically things like that. You will also get a C-loop file, as I mentioned previously, which is the file that you input into our loop browser. So this C-loop file allows you to look at 2D projections inside of loop browser. It allows you to cluster your cells, look at differential expression, and also you can do some interactive exploration inside of loop browser. So for example, clustering different cells together, looking at differential expression, and also projecting different clusters individually and all together. So this is something that I can also do a demo of if you want. But yeah, this is kind of further downstream of the wet lab portion. The Cell Ranger pipeline runs either on a local mode or a cluster mode, so depending on what your institute provides. In the local mode, it runs on single standalone Linux system and only Linux. Take note of that. It requires 64-bit operating system, this or Ubuntu, and it requires A+ cores, 64 gigabytes of RAM at a minimum. Otherwise, it can be run on a cluster mode, and the requirements are listed here. To download Cell Ranger is very easy. There's a set of instructions on this link here. Again, I will send these slides to you so you can look at it yourself, download it yourself, and the installation is quite easy as well. You can also Cell Ranger automatically produces a diagnostic file for all of your analysis runs. And if you encounter any troubles or run into any issues, you can send us this diagnostic file, and our in-house bioinformaticians can take a look at it with you. So we can kind of help you, troubleshoot any runs that may have had any issues. And you can send it directly to us from using this command line here, or you can even email support@10x Genomics for any issues, not just bioinformatics, that you have. Okay, so training next steps. So usually I would go through some of these items about kind of what samples you would run, whether you would be generating featured barcode libraries or not. But since we've already kind of organized a couple of wet lab training sessions, I won't go through this, but so we already have kind of volunteers that have their own samples that we're going to run. But just know that these are kind of some of the checklists of things that you need to kind of think about, and and that you can also discuss with me as well if you'd like to do your own wet lab run with your own samples. For the people that are attending the wet lab training sessions, the two that are coming up, since we already have people that are providing their own samples, what we will actually do in these sessions with the people that aren't providing their own sample is to run a training kit. So in order for you to get some hands-on experience with loading the chips and running the instrument. Obviously, the people providing the samples want to handle their own samples and probably don't want other people handling them. So in order to do this, we will practice loading a training chip using emulsions, generating emulsions and recovering gems without any cells or nuclei in them. So basically just using PBS or water. This training chip includes mock kind of microfluidic chips. Well, sorry, not mock microfluidic chips. They're real microfluidic chips, but that you will be loading mock samples and mock reagents onto, including gel beads. So you will still generate an emulsion that you can then break and recover, and we will go through all of that while the people providing the samples are prepping their samples. So there is a protocol for this, and I will be sending this out to the people attending these sessions. But just note that this particular protocol for the training kit is specific to the training kit and has kind of different volumes and everything to the actual real kits that you will use when you run real samples. And these training chips and reagents are not compatible with the real samples. Just take note of that. So that's kind of it for the site prep. So there are a lot of support materials available on our website. You can also email support@10x Genomics, and also email me. There is the support site where you can find lots of user guides and protocols for different sample types. We also have a Q&A knowledge base where you can, where we've published a bunch of articles kind of describing specific questions that people have sent in. So kind of FAQs. So you can take a look at that if you have any specific questions towards, like, for example, specific sample types or specific assays. And of course, you can always email support@10xgenomics.com as well, or email me. We will go through the workflow in a lot more detail in the next session. So yeah, we'll go into some more details. If you have any questions that you want to ask then, you can also leave them until then as well.